Alrighty, future biologists. We're back with another video. Let me make that a little bit brighter if we can. There we go. All right, so we're back with another video. This time we're going to be talking about dihybrid crosses. So, as always, if you don't have this lovely handout, please make sure you come find me and get this blank one so you can watch this video if you missed out on this lesson in person, or you can just use notebook paper from your book of biology. So by now we've already looked at monohybrid crosses, which was a cross between for one trait. So today we're looking at dihybrid crosses, and dihybrid meaning two. So this is a two trait cross. So in our example today, we're looking at a cross between eye color and we're looking at hair color for two people. So this is where it gets a little fun. So with a dihybrid Punnett square, you're going to have not one, not two, not three, not four, but 16 boxes. Yes, you heard me correctly. 16. So each genotype will have 4 letters. And we're going to walk through a process called FOIL to help you remember how to do this. So there's going to be 2 letters for each trait. So FOIL is for, you're going to do FOIL to figure out the gametes first. We're going to have more on the back of this. So, for example, we have big T is dominant because it's the uppercase for being tall, a lowercase t or little t for short. And then we have red is going to be for, let's say if we're going to do hair color. That's interesting. And then you're going to have big R represent dominant for red, little r be recessive for white. So here are what our genotypes, the code would be for each one. So if we were looking at a tall red plant in this example, a tall red plant, tall is going to be dominant, so big T, and red is also going to be dominant, so we know it's going to have a big R. So all of our possible genotypes we would get. First, we know big T would get us tall, and big R would get us red. But is that the only combination that we could get this from? No. We could get big T, and big R and little r. But does the fun stop there? No, it doesn't. We can go big T, big R, and of course, big T, and big R. So these are all the possible genotypes that we could get for a tall red plant, okay? And we're going to take a quick break here in 15 seconds for a quick pause for a word from our sponsors. All right, students, and we are back. So we've completed our first example. If we wanted a short red plant, well, we know short is recessive. So what do we need in order to get a short plant? You need 2 T's, right? 2 little t's. So we know short is recessive or weak. So we know we need 2 little t's, and we still get a red plant, so which is going to be dominant. So we know it's going to have a big R. So what are the possible combinations? Well, there are only 2, right? You can have little t, big R, or you can have little t, big R, little r. Okay, let's move on to our next example. Tall plant, dominant, big T. Recessive will be our white plant, recessive. So it has to have 2 T's. So our combinations are big T, little r, little r. Then, of course, big T, again, little r. Our last plant, or our last example, we're going to have a short white plant. So we're going to go recessive, because it's short, little t, recessive for white plant, and it's going to be little r, little r. Our only combination for this, little t, little r, little r. So this first genotype tells us what the height would be, and our second set of letters tells us the color. Now, we're going to flip it over to the back to figure out what we call FOIL. So, FOIL stands for, and this is how you FOIL for your gametes. It is first, outside, inside, and last. So, as we walk through this example, I will have some different colors that I will use to kind of make it a little bit easier to see how this process works. So, for my first example, I have big or little h, and then big T, big T. So my first thing I'm going to do, so because this H is first and this H or in this T is first in its section, I'm going to go first here. So my first combination for the gametes is going to be little h, big T. Next, we're going to go outside. So switching colors here, I'm going to underline this because I did first in blue. Outside is going to be in green. For outside, I'm going to take here, come outside for little h, big T. Next, we're going to go inside. So the two inside letters, which would be these two here, I'm going to get little h, big T. And then last, because of these two h's, this one's last. And we're going to do it in red for last. And then here. So my four gametes would be here. Now, we're going to follow the same process with this parent. So we're going to stick with our same colors, blue for first. So here, I get big H, big T, green for outside, here, big H, big T, pink for inside, little h, big T, and then red for outside. I'm sorry, red for last. These are our two last. Here, we're going to have little h, big T. Okay? So, it's important to note, I know this may seem confusing. We're going to go over this today. We're going to have a practice example coming up next in the next video, but do not stress about don't stress about whether or not you get dihybrid Punnett squares. Because on your quizzes or tests, on your test, your Unit 7 test that we take on February 3rd, the dihybrid chart will already be made for you. So that's one less thing you have to stress about. The boxes will already be filled in. You may have to fill in one or two boxes, but that's it. And excuse me. And as far as your EOC goes, they don't ask you a lot of dihybrid questions. It may be one, maybe. That's a big maybe. So you want to know how it's done, but don't stress out about it if you get confused. And of course, you can always rewatch this video if you need to. So we're going to follow the same format again for our next example. So we're going to go first. So this is the first letter of the R's. This is the first letter of the Y's. So my first here, and we're going to get big R, big Y. Outside, we're going to come here, and we're going to get big R, little y. We're going to go inside and get here, big R, big Y. And then for my outside, or my last, I'm sorry, I keep saying outside twice. For last, we're going to go here, come up, our last, and we're going to have big R, little y. We're going to cross that with now little r, little r, little y, little y. So again, start with my first, boom, little r, little y, outside, little r, little y, inside, here, little r, little y, and then last, here, little r, little y. Cool. Makes sense? Got it? Let's do one more just to make sense. Just to make sure we got it. So now we're crossing big S, big G, with little s, big G, big G. So we're going to go first here. Gives us big S, big G. We're going to go outside here. Gives us big S, little g. We're going to go inside. Gives us little s, big G. And then we're going to go last out here for little s, little g. Now, I know some of you may be wondering, Mr. Scott, I thought dominant letter always went first. That is true, but this is not going to stay as our genotype. So this is just the first part of it. Once we walk through an example, I'll show you why, and then it will make a little bit more sense. But for foil, this is how we get done. So whatever the first letter is, so when we're on inside, if it's little s, big G, that's how you're going to write it. Okay? Let's do this last one. Starting off with our first, little s, big G. Going to our outside, little s, big G. Going to our inside, little s, big G. And going to our last, little s, big G. Okay? So here are steps to solve a dihybrid. So first thing is you're going to get your gametes. You're going to go foil, just like we did here. You're going to put each parent's gametes on the top and on the left, just like we normally do. So each one will have 2 letters. Next, you're going to fill in the boxes. Now, inside the boxes, we're going to have 4 letters. The last step, you're going to analyze the result. So what I need you to remember from this, we're going to go first, outside, inside, and last letters. Man, I cannot spell today. And last. That's supposed to be inside. All right. So there's that for you. Hopefully it wasn't too much. I'm breaking this up into 2 videos because it can be kind of wordy. Check out the second video where we walk through a live example using this format. Until next time, I'll see you in the halls.